Hi everyone, welcome back to your channel Clarity for Sure and Agriclarity. Today we are back with the next sessions on the MCQ practice for this year's IBPS AFO mains examination. In today's session, we'll be discussing the sericulture and lac culture related questions. Okay, so before moving on to today's class, I want every student watching this session to go to our channel Agriclarity, where you can find the playlist regarding the PYQ analysis of IBPS AFO examinations from the year 2021 to 24. While all the entomology, particularly if we say particularly about the entomology, I have given 20 from the year 2017 to 24. 2024 question analysis of the year-wise analysis, the topic-wise analysis. What are the important topics you should see? What are the topics you should skim? So, if you have seen these all videos, you'll be able to understand why or what is the importance of today's session holds. Okay, so please go and watch the session and do let us know how you felt, how was that particular analysis video for you, so that you can have a streamlined preparation for this year's examination. Okay, so let's start with the first question of today's session. Which product is referred for coating fruits and vegetables to prolong the shelf life? Okay, so here are the products which are related to the lac culture. Okay, so the options are lac dye, wax, de-wax, bleach shellac, shellac, then elatic acid, and raw shellac. Okay, so the answer here is C, which is de-wax, bleach shellac. Okay, why? Because these are the wax-free, as the name suggests, hard and shiny, making it ideal for the edible coating of the fruits and vegetables, extending shelf life without affecting taste or appearance. Okay, so what we can say that the de-wax bleached shellac is actually a edible kind of shellac. Okay, so we can conclude from this option that it is a edible kind of lac. Okay, shellac, byproduct of lac. Okay. Moving on to the next question. The key advantage of de-wax bleach shellac over regular shellac is what? Okay, you have to differentiate how de-waxed and regular shellac is differentiated. So the option says that they have higher solubility in water. Okay, there is lower melting point. Then next, they are harder, brighter, and completely free from wax. Then they have reduced adhesive properties. And next, the increased toxicity. As in the previous slide, we have discussed the answer. Again, here is C, because the de-waxed bleach shellac is harder, brighter, and wax-free, making it suitable for use in the cosmetics, food coating, and pharmaceutical applications requiring a smooth and shiny finish. Okay, so from these two questions, you can see how the byproducts of lac culture and the questions can be formed from the byproducts of the lac culture, right? So in this way, you have to cover the lac culture subject. Okay. Next is which disease affects the silkworms. Now we have moved on to the. I have tried to put mixed questions from lac culture and sericulture so that you get accustomed to how to attempt these kind of questions, right? Then the question says that which disease affects the silkworms during high humidity and temperature, causing a blackish, fragile larva skin in non-mulberry silkworms. So here is the from the pest and diseases topic of the sericulture. Okay, so the options are viruses, microsporidiosis, mycosis, bacteriosis, or muscardine. Okay, so answer here is B. A, the viruses, which is also known by polyhedrosis, and is which is a it is a problem in non-mulberry silkworms, which is a viral worsened by high humidity and temperature, causing a blackish fragile larva. Okay, so in this way, we have seen how the pest and disease related questions can be framed. Okay, moving on, the next is the disease that show irregular hatching of eggs, wrinkled larva skin, and the black pepper-like spots is characteristics of muscardine, Fletchery disease, aspergillosis, grassery, या फिर pebrine. Okay, so you have to choose which disease symptomatic or symptomatic characteristics are mentioned here. Okay, so answer here is actually pebrine, which is a Nosema bovis, which causes these kind of symptoms and can be transmitted through eggs. Okay, so here from here we have revised the characteristic features of the pebrine disease. Okay. Next is which of the following describes the volatinism of the silkworms? One of the most important topic of sericulture, volatinism. So what is volatinism? It is the number of cocoons produced per larval stage, or it is the number of generations of broods produced by the silkworms in a year. It is the number of larval molts before the pupation, or it is the lifespan duration of the adult moth, or it is a duration of reeling process. So it is very clear. It is the number of generations of broods produced by silkworms in a year. Okay, so if we see what is volatinism, refers to number of broods generation per year. Univoltine means one generation per year. Bivoltine means two generation per year, and whereas multivoltine means several generations per year. Okay, so in this way we have discussed the three types of volatinism here. Okay. Now we have to choose the incorrectly match regarding the non-mulberry silkworm and their silk type. Okay, we have to choose. You should be familiar with the scientific name also. Okay, from all the silkworms of the the non-mulberry silkworms. Okay, so if you are familiar with their scientific name, you can easily differentiate the options here or eliminate the options here, right? So the answer here is actually D. Okay, D, which is the cone silk. Okay, it is actually the Philosemia resini is actually a eri silk, not a cone silk, which is actually the cone silk is produced by Paikypasa octus. Okay, and that is why the D option is uh, incorrect here. Okay, right. Okay. Moving on to the next question, the question is saying that how the Silk Route was connected with China. Okay, how so it is generally kind of a we can say a GK kind of question, but it is very important from the sericulture perspective. Okay, so the Silk Route connected with China from where? Africa via Indian Ocean, Europe via Pacific Ocean, Japan through Manchurian corridor, then Arabia through Silk Bay, then Mediterranean region through Central Asia. So answer here is actually the Mediterranean region. Okay, the Silk Route was connected with China through the Mediterranean region. Okay, so the Silk Route linked with Eastern China to the Mediterranean region through Central Asia, facilitated trade in the silk, gold, and ideas. So that is why the E option is correct here. Okay, so you can see how the questions can be. What you can expect from these two topics. Okay, the sericulture and the lac culture. Moving on to the next question. Here we have to choose which characteristics describes the sexual dimorphism in the lac insects. Okay, so it is very important to know the biology or the identification characteristics of the lac insects as well as the silkworms and the non-mulberry silkworms. Okay, so that is why I have tried to put here these kind of questions. Okay, so options are: the male is larger than female, it is wingless with well-developed antennae. Then male is smaller than female, red in color with wings and reduced eyes and antennae. Then female is smaller than male, pyriform and has wings. Both male and female are smaller in size, but male is winged. And last is female is red while the male is yellow. So correct answer is actually the B option. Okay. Why? Because the male lac insects are small, ranging from this 1.2 to 1.5 mm. They are red in color, have hyaline wings, reduced eyes, and degenerated entity, which is important point here. Okay, you should be very familiar with this point. Okay, it may highlight it. Okay, so the male are generally they have hyaline wings, reduced eyes, and degenerated antennae here. Okay, while the females are larger here, ranging from 4 to 5 mm, they are piriform, but their winglets are have degenerated antennae and legs as well. Okay, so there is the dimorphism in this lac culture is very extreme, reflecting that there is different roles in different. These two species, these two entities have different roles in reproduction and lac secretion. Okay, so that is why this was very important question. So I have tried to I have framed here. Okay. Moving on to the next question. How many crops of lac can be obtained in a year for Rangini strain and during which month? So again, the lac strains and their cultivation is one of the important topic of the lac culture. So you should be familiar with the all the strains, Rangini strain and Kusumi strains. Okay. So here option says that one crop June and July, one crop October and November, two crops June, July and January, February. Again, two crops Kartiki October or November, Baisakhi, May and June, then three crops January, February, May, June, and or October or November. So the correct answer here is actually D option, which is two crops. We can take two crops during Kartiki and the Baisakhi months. Okay. So Rangini strains are generally Rangini strains yield two crops annually, and we have said all the months which are located, which are specific for these two crops to rear. Okay. Next, we have to choose the options which correctly describe the duration of the lac insect life cycle. Okay, one of the important topic. I have tried to give biology related questions of the lac insects here. So you have to choose how many months duration of the what is the approximate month of lac insect life cycle. So answer is D, which is 6 months. Okay, nearly half year. Okay. So the entire life cycle of the lac insect is about 6 months, allowing two crops per year. So in this way, we have revised that two crops can be taken annually and they can complete their life cycle in 6 months. Okay. So in this way, we have revised important points of the lac culture. Moving on, we are this questions is again targeting lac cultivation. The main purpose of artificial inoculation in the lac culture is generally there are two types of inoculation types, and one of it is artificial inoculation. Okay, so you have to choose what is the purpose of artificial inoculation here. So it allows natural infection of host plants by nymphs. It prevents the lac insect from producing lac. It ensures uniform inoculation and overcome drawbacks of natural inoculation, to harvest immature lac only, to grow lac on non-host plants. So the answer here is C option. Why? Because the artificial inoculation is actually done manually to overcome the limitations. Okay, it is generally done to overcome the limitations here of the natural inoculation. Then only you can go for the artificial inoculation, such as non-uniform spread, weak host nutrition. Okay, so by pruning the host crops, by pruning the host plants and tying the brood lac to fresh twigs again, uniform growth and better yield can be ensured. So it is kind of, this inoculation can be used as a aid to overcome your yield loss yield you have faced. Okay, so that is why in that way artificial inoculation comes in rescue. Okay, moving on. Next question is which of the following inoculation types occurs naturally when swarm nymphs infect the same host again? Again, we are going with the two types of rearing process which we have discussed earlier. There is a artificial inoculation process and there is natural inoculation process. Okay, so you have to choose the second type here, which occurs naturally. Okay, there is self inoculation, artificial inoculation. Artificial inoculation is easily eliminated here. Okay, then brood inoculation, pruning inoculation, and mature inoculation. So answer is actually A here. Okay, the natural inoculation is also known by self inoculation process. Okay, so that is why the natural inoculation occurs automatically. That is why it is named self inoculation, where the nymphs settle on the same host. Its drawback includes that there is non-uniform spread and poor host nutrition, making the artificial inoculation preferable here. Okay. So in this way, we have discussed the inoculation types in the lac culture. Moving on, the next is inoculation period for the Kartikeya crop of Rangini strain is in which month? Okay, if you have seen if you have carefully seen the months for the Kartikeya crop, okay, so you can easily guess the inoculation period here. Okay, the options are January, February, May, June, July, October, November, and February, March. So answer is here is C option, which is June, July. Okay. So it is the first, the Kartiki is actually, it is inoculated during June, July, whereas the Baisakhi was inoculated in the October, November. Okay. So I hope from this question, you have revised what are the inoculation months of Kartiki and Baisakhi. Okay, so this question is very important. Okay, and in this way only the examiner can ask the questions regarding the lac strains. Okay. Next is, which of the following practices helps in reducing the brood lac loss, again regarding the pest and disease of sericulture, and ensures the quality production here? I mean, yes. Oh, sorry. It is targeting the brood loss of the lac culture. Okay. Then the questions options are harvesting immature lac for all the crops. Okay, using the same host plants for multiple years, harvesting mature lac within 7 to 10 days before the nymphal emergence. Okay, then avoiding pruning before the inoculation, and lastly ensuring brood lac in humid conditions. So here you have to choose the actual practice which help in reducing the brood lac loss. Okay, you can also get the required lac, and as well as you also resist from any loss of brood for the next rearing season. Okay, so in this way the answer actually option C is correct here. Okay, so why? Because a mature crop should be harvested 7 to 10 days before the nymphs emerges. Early harvesting may kill nymphs, but late harvesting may allow nymphs to escape. So that is why reducing the brood lac available for the next inoculation. So that is why they are uh, they are harvested 7 to 10 days before. Okay. Moving on to the next question, what is the ideal length of the brood twigs? One of the important question, and I have feeling that it can come in examination if there is any question from the lac culture. Okay, so what is the ideal length of the brood twigs used for the artificial inoculation? 20 to 30, 50 to 60, 5 to 10, 40 to 50, or 10 to 15. Okay, so answer is 20 to 30 here, the option A. Okay. Option A. Why? Because it's the optimal for tying a fresh tree twigs, ensuring sufficient gravid females for successful inoculation without causing any handling difficulties. So it is one of the important range or length of the brood twigs to be selected for artificial inoculation. Okay. So 20 to 30 is the ideal length here. Okay. Please make note of it. Next is oozy fly. Okay, one of the important fly of, causing the pest and disease problem in these sericulture. Okay, Exorista bombycis. Okay, it inflicts the maximum damage to silkworm cocoon crop under which of the following conditions? Okay, what are the favorable conditions to have a maximum damage from this fly? Okay, so option says that winter season in the temperate regions, rainy season in the tropical or silk producing states, dry season in hilly regions, and summer season in arid regions, and lastly cold months in the north region, North India. Okay, so the answer here is actually option B, which is the rainy season. Okay, so UZ fly occurs throughout the year, but it is very severe during the rainy season in Karnataka, Andhra, and Tamil Nadu, or we can say the South Indian side, South India side. Okay, so where the premier silk, where is this premier silk producing states? Okay, so that is why the B option is correct here. So you should be familiar with the favorable conditions of the oozy fly to have maximum incidence. Okay, the rainy season. Next is before reeling, the cocoons are conditions for. So before reeling on, the cocoons are conditions for. Options are 1 to 2 days, 3 to 5 days, 1 month, 15 to 20 days, and 7 to 10 days. So your answer is 15 to, yes, sorry, the 7 to 10 days. Okay, so 7 to 10 days. Option here is last option E is correct here. Why? Because it improves the silk reeling quality. So that is why they are conditioned and the cocoons are dried for 7 to 10 days. Okay. Next. So during the spinning process, the silkworm first produces. What is the first structure, or what is the first product which is produced by the silkworm here? Okay, so there is a cocoon proper, there is a pupa sheath or hammock structure, the floss or sericin threads. So actually the answer here is hammock structure. Okay, what is hammock structure? The silkworm spins a loose hammock first, providing an anchorage for the spinning prop for the cocoon. To have an anchorage for the cocoon properly. Okay, so this structure is known by hammock structure. Okay. Next is, there are three main regions of silk glands in Bombyx Mori, which are you have to choose a main. Again, I have given the biology related questions also. So you have to choose the three main regions where the silk are produced or where the glands are situated. Okay. So fibronine, sericin, and max gland, posterior, bulbous, and spinnerate, basal, medial, apical, posterior, middle, anterior, and none of this. So answer is actually the posterior, middle, and anterior. Actually, the question is asking the regions. So the regions are posterior, middle, and anterior, while the option A is actually the the product. Okay, the production of particular regions. Okay, secretion of particular regions. Okay, the posterior secretes the fibronine, the middle secretes the sericin. Okay, and the anterior secretes the wax gland. So in this way, the silk gland consists of posterior, middle, and anterior regions, which is specialized for secretion of fibroin secretion, sericin secretion, and the transport respectively. Okay, so in this way, we have seen the main regions as well for the silk of the silk gland. Okay. Next is two silk filaments, which are called brins, secreted from each silk gland combine to form. When these two silk filaments or brins combine, they can the entity formed is known floss, bave, cocoon shell, sericin gum, or fibroin thread. Okay, so answer here is B. Actually, bave. It is called bave. Okay, so each gland secretes one filament brin. The two brins unite, glued by sericin, forming a single wave, or we can call it as silk thread. Okay, so silk thread is also known by wave, and the silk filaments are known by brin. So from here we have discussed the important terminologies also. Okay. Next is the silkworm ceases feeding and begins cocooning after about. You have to choose an instar after which the actual cocooning process is it begins. Okay. So the fifth instar, third instar, fourth, second, or immediately after the first four. So generally, the fifth instar is the correct answer here. Okay. So the fifth, after the fully grown fifth instar larva stops the feeding, empty their gut, and begins feeding in 7 to 9 days. So if we have given the instar as well as the day, the period after which the actual cocooning begins. Okay. Fifth instar and how many days? 7 to 9 days. Okay, right? Next is, okay, so that was it. Okay, for more such questions, you can do check our Telegram channel, Agriclarity, where we have posted such kind of questions as there as well. You can get all the questions regarding the entomology, sericulture, lac culture. Just before this day, I have also posted apiculture related questions on the Telegram channel. So do check our Telegram channel where you can get this actual examination kind of examination pattern kind of questions there. Okay, so that was it. I hope this session was helpful. If you have any doubt, any particular topic you feel that we should come up with the detailed video on that particular topic, please let us know in the comments, and also let us know how this session was for you, because uh, in some way or other this session would be helpful for AFO examination, or else other agriculture examination, right? So do let us know your suggestions or feedback in the comments below. And that was it. Thank you and have a nice day.